Assalamualaikum and good day to all. So, welcome to the APBioNETTalk 7. Okay, the title today is Uncovering Increasing Arbovirus Outbreak in the Subtropical South America through Genomics and Bioinformatics. So, the speaker today will be Dr. Gabriel Wallau. Dr. Gabriel Wallau is a public health researcher and a principal investigator at the Oswaldo Krauss Foundations and Bernard Snott's Institute for the Tropical Medicines. He coordinates several projects focused on genomics of pathogens and vector. One of these projects track mosquito-borne pathogen outbreak that are spreading their reach to subtropicals and temperate regions of the planet using genomics and bioinformatic tools. He published several studies focused on the molecular epidemiologies of the arbovirus in the Brazil and South America and their impacts on the Brazilian population across the country. Without further ado, please welcome Dr. Gabriel Wallau with the talk.
Thank you very much for this kind introduction. So I think I can try to share my screen here. Let's see. Okay, can you see my screen? Okay, so I hope you can all listen to me well. So first of all, I'd like to thank you very much for this opportunity. I'd like to congratulate for these online talks. I think that's amazing. Amazing learning from the pandemics that we can all connect across the globe to share experiences and so on. So I think this symbol of the APBioNETTalks is amazing to connect the dots around the globe. So again, congratulations for this initiative, and I'm glad to be here speaking a little bit about the work we have been developing inside of my group. So as I just said in this nice introduction, I am a public health researcher from the Oswaldo Cruz Foundation, and I also have an affiliation with the Bernard Institute for Tropical Medicine in Hamburg, Germany. And today I'm going to talk about some of the work we have been doing characterizing arbovirus outbreak in subtropical, but you're also going to see that's not only regarding subtropical, because to understand the whole picture, we need to understand where those viruses are circulating in other parts of the country and the world. But that I will give a special focus on genomics and bioinformatics, focusing on the tools and solutions we have been developing to track those viruses. Okay, so let me see here. Those are the topics I will address today. I'm going to talk about arboviruses, give you some background about it. I will also talk about how impact human arboviruses, give you some context on arboviruses in Brazil and knowledge gaps, how we are filling the gaps of genomics and bioinformatics, and which are the opportunities and challenges regarding this topic, and how we can convert this knowledge on preparedness and maximum impact on public health. So to give you a little bit of background regarding arboviruses, arboviruses are arthropod-borne viruses transmitted by arthropods. The main vector are mosquitoes, mites, and also phlebotomines. But among those arthropod vectors, mosquitoes are recognized as the most important ones. And here in this slide, we have one of the main vector of arboviruses across the globe. So in the last part of the slide, you can see Aedes aegypti and Aedes albopictus. Those are the highly anthropophilic mosquitoes that transmit arboviruses to humans, as I mentioned, across the globe. I'm going to show in the next slide. But with this slide here, I would like to highlight that arboviruses can have these three main cycles. One of them is urban epidemic cycle, which is mediated by those highly anthropophilic mosquito species in the urban environment. And humans are not only the main reservoir, but also the main impacted hosts. And here we have some examples like chikungunya virus, dengue virus, Mayaro, yellow fever, and Zika virus. I'm going to talk more about this virus later on. So we also have an enzootic cycle. Those enzootic cycles, those arboviruses they infect non-human primates, they infect rodents, and they also infect birds as the main reservoir and host infected. But eventually, those mosquito species, which are not, most of the time, are not Aedes aegypti and Aedes albopictus, or other sylvatic mosquito species, they can feed on humans and transmit those viruses to humans. You can see that here we have basically almost the same viruses as here, because that was that's the main hypothesis to explain the origin of those viruses that currently do have an urban epidemic cycle. So all of those viruses emerged from an enzootic cycle in which they infected, and they probably continue to infect sylvatic animals in the wild. But then, because of bridge vectors and the approaching of the human population to those areas and vectors, the virus was transmitted to human, and then the virus was established in a urban epidemic cycle, being mediated by Aedes aegypti and Aedes albopictus. And we also have episodic cycle. What's the difference of episodic cycle of arboviruses? Here in this case, the main reservoir and amplifying hosts are domestic animals like swine and sheep, right? Here we have two examples, JAV and RV-FV virus, in which they circulate substantially among domestic animals. And because we humans do have a very close relationship with domestic animals, then those mosquitoes here can feed on sheep and swine and then transmit the virus to humans, okay? But in this case, there is a particular difference here because although humans are highly impacted by JAV and RV-FV, they suffer from the infection. Humans do not generate high enough viremia to allow a new mosquito to get infected from human and then keep on the transmission. That would be the requirement to establish a human transmission cycle, right? So most of the time, humans here are dead-end hosts, although they suffer from the infection, but the main cycle here is maintained through domestic animals and mosquito vectors. So going on with talking more about high impact arboviruses, here we have a map from 2018 from a review paper showing the distribution of those high impact arboviruses. You can see dengue here. Dengue is spread all over the globe. Of course, mainly in the tropical region of the globe, because here we have the largest population of the vectors, right? And the conditions for this vector development and transmission of arbovirus consequently. We also can see here that more recently chikungunya virus also reached all tropical regions of the globe. So those are the two arbovirus with highest impact on the human population. But we also have some other very important viruses like yellow fever virus, which originated in Africa, is still circulating in Africa, but also circulates here in America, causing outbreaks every 3 or 4 years. We have Mayaro virus or push virus, Zika virus, although Zika virus is a bit outdated here because we know that Zika virus is continually circulating in Asia as well. And we also have JAV here in Asia and Southeast Asia, which also outdated because this year we had several cases of JAV in Australia, right? But that's the current understanding, more or less the current understanding about the distribution of those high impact arbovirus in the globe. So as I just mentioned in the previous slide, those viruses are mainly restricted, or they circulated more in the tropical region of the globe, right? In tropical countries, they do have a high impact on those populations. In this paper here, some years ago, in collaboration with some other researchers from Fiocruz, we revisited all the genomic data from dengue, different serotypes of dengue virus, and also genotypes. But we also reviewed the current find at that time about chikungunya and Zika virus. And what we found is that there is a common root of viruses, of arboviruses, that spread through the Pacific Islands, from Asia to the Pacific Islands, and then those arboviruses reach Central America and the Caribbean islands. And from those countries here, then they spread through the South and North in the Americas. So that's a kind of well-known route of arboviruses spreading across the globe, and particularly focusing on South America, one of the main routes in which they arrive. Okay, so of course it's important to highlight it's not the only route. We also can see here some other, and there has been cases reporting direct transmission from other countries to Brazil, for example, but that's a common route we found revisiting all this genomic data at the time. So focusing now on arbovirus in Brazil, which the main topic and where I work mostly, where we have more data. In Brazil, the first cases of dengue virus date back to 1800, 1923 in Rio de Janeiro. And then in 1986, several outbreaks of dengue virus have been detected across the country, right? Showing that the dengue virus was spreading and reaching all corners of Brazil at that time. And here we have some figures of a more recent paper we published in Nature Communications some years ago, showing dengue cases since 2001 until the Zika virus emergence in Brazil, right? So our goal in this study was to understand what happened when Zika arrived in Brazil and what happened after that. Why Zika cases dropped significantly after that, right? So, but here with this slide, I'd like to show you that dengue is hyper endemic in Brazil, like in many other tropical countries across the globe. We have huge waves of infections, but something happened here when Zika arrived, right? So here in 2016, we had triple epidemic, what we call triple epidemic, because we had, again, the dengue virus infection wave, but Zika arrived, and then we had a huge peak of Zika cases. And at the same time, we had here, it's another graph here, we also had the chikungunya, huge wave of infection, because chikungunya arrived in Brazil a little bit before Zika virus. Then the three arboviruses, which are transmitted by Aedes aegypti, established this urban cycle and infecting 1,000 million of people. And our idea with this paper here was to understand, well, what happened with dengue and mostly Zika and dengue, and what's the relationship of those arboviruses after the triple epidemic? Because after the triple epidemic, we can see here a very low number of cases of dengue virus and also Zika virus. And that was not particular for only those arbovirus, but it was also seen for chikungunya virus, a much lower curve of infection, smaller curve of infection was detected. So what happened here after the triple epidemic? So give you a little bit more context regarding arbovirus in Brazil. Here we have some historical maps that help us to understand the situation today. Here on the left, we can see a very outdated map from 2007, where people cataloged the abundance and presence of Aedes aegypti in Brazil. And here it's a more, it's covering wider range, period range in Brazil, considering dengue cases, and we can see where most of the dengue cases were concentrated, right? Two interesting facts here is that we can see two barriers. One barrier here, where Aedes aegypti was most well distributed in this part of the country, and another barrier here in the Amazon rainforest. So the Amazon rainforest was, let's say, buffering the spread of Aedes aegypti, and it makes sense because Aedes aegypti is a urban adapted mosquito, right? So it's not well adapted to large forested areas, although the ancestral species of Aedes aegypti in Africa came from forested areas, right? It's important to highlight it. But Aedes aegypti in Brazil, we had the introduction in the past of the urban adapted mosquito, and then it makes sense to have this kind of buffer here because of the Amazon rainforest. But here we have another context. In the south of Brazil, we have a subtropical region, right? So although everybody thinks when Brazil came to your mind, I'm quite sure about that, we think about, well, it's a tropical country full of beaches and beautiful country, of course. But most of the time people don't think that we also have a huge variation in climate across Brazil. And here in the south of Brazil, we have a subtropical region, right? Which here in the right side of the slide, you can see the temperature variation throughout the year. So the x-axis is the whole year from January to December. And here in the y-axis, you can see the years from 2005 to 2020. And this position of those squares here follow the states distribution in Brazil. So here in the southernmost part of the country, you can see well demarcated seasons when in the middle of the year, like from June to August, we have very cold winter, reaching sometimes negative temperature in the southernmost state of Brazil. But that's very clear for these three last states here, the southernmost states of Brazil, right? For all the other states, it's more or less tropical climate with some variation, of course, with some more well demarcated season and not so well demarcated season. Here in the northeast part of the country, what we can see more is like humid and dry climate, not well demarcated season like we see in the south. But why I'm highlighting this here, because this kind of well demarcated season and clear fluctuations of temperature, humidity, and so on are exactly the cause of buffering Aedes aegypti expansion to the south of Brazil. And also, of course, if the vector is not there or it's having more hard time to reach the south of Brazil, then we have much less arbovirus infection in that part of the country. But the problem is, as you all know, global warming is here, climate change is here. And when we look at recent data from 2021-22, where Brazil reached the largest number of dengue cases from the whole history, the record cases, we can see here the number of municipalities in Brazil which registered dengue cases in that period of from 2021 to 2022. And we can see that many of those municipalities showing dengue cases in more recent years are in the southern part of Brazil. And that's the question in here is why we are seeing this increase in dengue burden in the south of Brazil, because now we do not have more well demarcated season in the south of Brazil because of all these climate changes. And Aedes aegypti and also Aedes albopictus are well distributed all over this region here. So basically, the barriers that existed in the past are being eroded with time. Okay, we're going to talk a little bit more later on. Now I'd like to bring to your attention the case of chikungunya virus. I will highlight some points here I mentioned briefly before. Dengue virus is endemic of Brazil since decades, more than 30 years. But chikungunya arrived in 2014, 2015, right? And when chikungunya arrived, we already had better tools for monitoring, for molecular detection, and also genome sequencing. And we as a community, were able to map the arrival of chikungunya here. In this first paper, for example, some authors show that chikungunya arrived in Brazil and South America as a whole through two different routes. One of them through the Central America and Caribbean islands to the north of Brazil, right here. But another lineage arrived here in the northeast part of Brazil, in the Bahia state. So the interesting thing here is that two lineages arrive basically at the same time in the country. And in this paper here, the authors also evaluate how likely those two lineages would spread countrywide and eventually would overlap in distribution based on the transmission suitability. That's an index that takes in consideration the vector abundance, the climate variation, and the impact on the transmission of those viruses in natural conditions. So using this index, you can see here that the Asian Caribbean lineage that arrived through the Caribbean and through Central America would spread countrywide, and that's not different from the CSA lineage, right? This one that arrived here in Bahia would also spread countrywide. But at that time, we only had sequences from the arrival here and some genomes detected here. So later on, I will come back to these points here to update you regarding the new findings we have. But now I would like to talk a little bit more about the bioinformatic tools, which allow us to then perform the studies I will show you later on. So here regarding bioinformatic tools, I'm going to address, I'm going to give you a broad overview about what we have been working inside of my group, and not only inside of my group, but also inside of what we call Fiocruz Genomic Network. So inside of my group, we started developing pipelines for bioinformatic analysis of viral genomic data back in 2015-16 when the Zika epidemic occurred, right? So we started developing the first bash scripts and pipelining several softwares to run very rapidly and generate some nice results. But then this knowledge was applied to different arboviruses. We studied chikungunya, we studied dengue virus, and so on. But then the pandemic came, and then because of the Fiocruz unit and several networks have been established at that time, we, as a Fiocruz, distributed all over the country with different units across Brazil. We joined together to build the Fiocruz Genomic Network. And because of my experience and my group experience developing pipelines and working with virus genomic data, we started to lead the bioinformatic part of the network, where we thought about the genomic surveillance engine, where we could, for example, enter sequencing data here from any kind of pathogen, of course, starting with viruses. And then we would be able to analyze very rapidly the genome of those pathogens. And then we could generate reports very rapidly, systematic reports to be shared with the decision makers. But we also were concerned about data protection, how we could ensure data protection, how we can share this information, not only with decision maker, but with also with the scientific community in a systematic way. So those were the three main pillars in which we focused to develop the tools I'm going to show you now. So and then during the pandemic in 2022, beginning of 2022, we published this paper where we discovered viral flow. So viral flow, again, the first script of viral flow we started developing it back in 2015-16, but it came about to be published in 2022, where we used SARS-CoV-2 example to show how viral flow would work and how it could be implemented. So viral flow is a pipeline for reference-based genome assembly and analysis. It has a public code. We were concerned of developing a pipeline that could be run in personal computer, but also we could scale it up and use it in multi-user servers. And then we decided to implement it inside of Dockers, inside of containers, to simplify things for the end user, to allow everyone to run the same analysis and to facilitate the installation of the whole pipeline. Because as you know, for those that work with bioinformatics since quite a long time, bioinformatic pipelines and bioinformatic tools overall they pose significant challenge for installation and usability. So using containers is one of the best strategies to allow not only others to use and replicate your analysis, but it's also very important to facilitate installation overall. So viral flow works using a reference genome. As you well know, we can use the first SARS-CoV-2 genome characterized from Wuhan, and then we map the reads against this reference genome, generating a bound file, a alignment bound file, and from this bound file, we can extract several data from each sample. It's also important to mention here that viral flow also has this model for filtering the raw reads, removing the low quality raw reads, removing adapters, removing primer sequences, and so on, using FastP. But then I won't go into details here, but I would like to highlight one of the major differences of viral flow compared to other pipelines, because as you also know, there are quite several other pipelines available that have been published before the pandemics or during the pandemics. Viral flow is set up to characterize not only a consensus sequence, meaning that the nucleotides that are supported by the majority of the reads will end up in the consensus sequence that's a majority-based consensus sequence for each sample. But also, we were concerned about the minor variants, right? Which are minor variants? What are minor variants? Minor variants are nucleotide sequences that are well supported. We have very strong filters to characterize minor variants, but they do not represent the majority of the reads. So those are truly nucleotide changes that happens during the replication of the virus in the host that should be characterized, because it gives us some very important information about the evolution of the virus, mainly about the intra-host evolution of the virus, right? And then with this characterization, we came up with one idea of reconstructing the majority consensus sequence and also the minority, a consensus sequence having the minor alleles in the sequence, in the final sequence, and then we could run these two genome versions from the same sample using the well-known softwares, right? Here is just a slide. I won't go into much detail, showing that the scalability of ViralFlow, we can run it in a personal computer. It usually takes from 5 to 6 minutes to run every sample. Of course, it depends on the size of the virus. It depends of the amount of reads you have. But we can also scale it up using HPC clusters, and we can process many samples at once when we can parallelize the ViralFlow run, right? And that everything I told you so far about ViralFlow was about the first version, 0.06, which was focused on SARS-CoV-2, right? So we only could run it systematically for SARS-CoV-2, although we could tweak it and use other viral reference genomes like monkeypox. We did it, but with a new version of ViralFlow, we'd like to implement some to make it more versatile, to allow ViralFlow to run for every kind of genome as long as we have a reference genome. So that was the next step. Then you can check in the github.io link here. That's the new web page of ViralFlow, and that's the web page of ViralFlow 1.0, in fact. So for ViralFlow 1.0, we decided to focus on singularity because Docker, we detected some issues regarding permissions to use Docker with the root permission inside of clusters, and then singularity was did not show this issue. And then we also, with the help of several people from our group, we decided to move ViralFlow to Nextflow. Nextflow is domain-specific language, specifically tailored for bioinformatics, I would say. It's not only used for bioinformatics, but it was developed from a bioinformatic point of view for dealing with large biological data sets, right? So here's the new version of ViralFlow. Here again, several of the steps are the same. Some new steps, we changed some softwares to improve robustness and speed. We also implemented other softwares like SnpF, which not only it uses the information, the nucleotide variation we detected to characterize if it generates amino acid changes or not, and if this amino acid change has more likelihood, is more likely to change something significant in the protein or not, right? But here I brought to you the main features of ViralFlow 1.0 that I would like to highlight as new features. The first one is that we made it more versatile. So as long as we have GenBank ID, we can pass the GenBank ID of this reference genome to ViralFlow, and then it will download the new genome and implement it to run at least the core analysis of ViralFlow for this new virus. So we already did that for chikungunya, for monkeypox, for rabies virus, for several other viruses. One important thing to mention here that ViralFlow is, even for the ViralFlow 1.0, it's more tailored for non-segmented viral genomes, right? Because for segmented viral genomes, we can run ViralFlow for segmented viral genomes, but then we will need to pass each segment at once to ViralFlow to run, right? Inside of ViralFlow 1.0, we can have these core analysis, which are agnostic models. They run for every virus, for example, characterize the intra-host variability, building 2 genomic versions for each sample. We can run SnpF for every, it's also an agnostic model for every virus. We also implement new functions to generate, to write in the folder only the reads that map it against the reference genome. And that's really important because even though irrespective of the protocol used to obtain the sequences, eventually, because we are working mostly with human samples, eventually we will sequence fragments of the human genome. And that's very sensitive data. So we need to be sure to share only the pathogen data, genetic data. And using this, we can be sure that all the reads generated in those files are only the ones that map it against the virus genome, and then we can share it safely without releasing any kind of data from the host, right? And we also have virus specific analysis here. Still, what we have implemented so far are the tools for SARS-CoV-2. But of course, we are always looking forward to implement new tools to also address virus-specific analysis for arbovirus, for example, or genotyping arboviruses. And here's something very that was implemented very recently in the ViralFlow 1.0 that we implemented the Bandash that was developed by Jonas Fuchs. And here in this picture is Alexandre, my PhD student that implemented it inside of ViralFlow. It's very interesting because this software here, developed by Jonas Fuchs, it outputs the genome coverage across the genome. So, and it generates not only a HTML file, but also a PNG or SVG file. So with this, everybody that's working with genomic surveillance and testing the protocol with divergent lineages can access if the coverage is dropping in some region, or if we can and should redesign primers to improve sequencing in a specific region of the genome. And here is just to give you an overview about the impact of ViralFlow. That was a recent analysis conducted by Felipe Desordi, another PhD student from my group, extracting data from Brazil and from deposited on GISAID. And this analysis was quite reassuring for our group, because we saw that from the sequences, about 130,000 sequences from Brazil available on GISAID, which also has the information of which software was used for genome assembly analysis. We saw that ViralFlow was the most used one, surpassing all commercial available software. So it was quite reassuring to see that ViralFlow has been used by the community intensively, and that it does not only output scientific valid results, but also that ViralFlow do have some impact on public health, direct impact on public health. And another tool I'd like to speak today, I need to check my time here, but is Viral Genome DB. That's a database we developed together with ViralFlow to not only host ViralFlow data, but also to combine this genomic data with metadata associated with each sample, right? So in Viral Genome DB, we developed it to facilitate several processes that were not automated inside the Fiocruz Genomic Network. For example, inside of DB, we have this dynamic visualization, and here we can see the lineages assigned by Pangolin coming from ViralFlow, of course, but one of the main focus here inside of the database is to evaluate the sequencing itself. So here we have the history of coverage of each genome per each sequencing batch of the network, that's also subdivided by different Fiocruz unities. And here is one sequencing run where we can see in the x-axis the coverage breadth of the genome, and here the coverage depth. So we can see if the sequencing worked pretty well or not so well to keep on with the analysis. Inside of VirusDB, we also implemented automatization for KeyState submissions. So now the user that entered the sequencing data and that launches viral flow inside of the database can also submit direct to KeyState with one click, and all the sequences that are above a given threshold, quality threshold, will be submitted directly to KeyState. And of course, as a last implementation, the user of this database can download all the data from metadata associated, but also all the data generated by ViralFlow, and in the end generate a standardized report. So this report was very, very important and still important in Brazil, because we share for every sequencing batch, we share this standardized report to the Minister of Health of Brazil, to the different state level and municipality level responsible for mitigation strategies. So inside of the database, we automated everything using Jenkins, which is a general orchestrator or for pipelining any kind of software. And then this is pretty nice because now we can easily transfer the high quality sequencer data from the network directly to KeyState. So after showing all those bioinformatic softwares and pipelines we developed, I'm going to talk a little bit more about the studies we conducted using, of course, these pipelines. Here is the paper we published this year, where we characterize the largest chikungunya outbreak in the southernmost state of Brazil. If you remember well, I told you the southernmost state, we had these well-demarcated seasons and temperature fluctuation throughout the year. And what happened is that before 2021, chikungunya cases had been reported, but never more than 52 cases per year. And most of those cases, they were clearly linked to people traveling to the tropical part of Brazil, the endemic region where chikungunya was circulating substantially, and then turning back to the state, right? So it was all kind of imported cases, let's say, but not inter-country, but interstate imported cases. But what we saw in 2021 that we detected 220 cases that surpassed all the cases detected in the previous years together in a single municipality, which is called São Nicolau. In this municipality, we detected basically a huge amount of community transmission. From this outbreak, we sequenced 23 genomes. All of them are from the CSA lineage, that lineage that entered Brazil through east, through the Bahia state here. And what we saw when we put all this genomic data together, we sequenced 23 genomes from the outbreak, from the 2021 outbreak. But we also sequenced several genomes from previous years. And that's perfectly matches our previous hypothesis. These years here, we detected isolated cases, imported cases from other states where chikungunya is endemic. And then in 2021, we detected a huge outbreak, showing that probably it was again imported in an index case from an endemic part of the country, but then in 2001, it was able to fuel an outbreak, a significant outbreak with enough community transmission. In the previous years, those imported cases were basically dead ends. There were no further transmission fueling outbreaks. But here in 2001, it was we clearly characterized it. So moving on to dengue virus, in Brazil, we detected now dengue 1, 2, and 3 from serotypes 1, 2, and 3. But basically, dengue 1 and 2 are the predominant ones, right? Here in the south of Brazil, the same southernmost state of Brazil, we can see the detection of Aedes aegypti per municipality. Overall, from 2018 to 2021, overall, Aedes aegypti is distributed all over the place, right? It's important to consider that here, 2020-21, it was 2 pandemic years, so the field teams did not really evaluate in depth the detection of Aedes aegypti. But still, with the surveys that were conducted, we can see that Aedes aegypti, at least in 2021, were distributed all over the state. And here we have a summary of dengue 1 and 2 detected throughout the years from 17 to 2022 in the same state. So regarding dengue, was detected in this southernmost state of Brazil since 2012. But again, following the same pattern I just described for you for chikungunya virus, most of the cases were travel related with very little communitary transmission. But here from 2017 on, we can see a marked increase in cases, right? Here we have a kernel in the log scale, showing that in 2022, which was the year with the highest number of cases in the state, dengue cases occurred all over the state, and a lot of cases occurred in some specific regions of the state. So, and to study this from the genomic point of view, we sequenced about 700 genomes, dengue genomes, both from serotype 1 and 2. Here we have a phylogenetic tree including the serotype 2 genomes. We detected 6 genomes from genotype 3 and 32 genomes of genotype 2. And one interesting finding of this preliminary data that we are still working on is that for genotype 2 cosmopolitan, where we sequenced 32 genomes, we detected 1 year overwintering, meaning that the same lineage that was present in the past year was fueling new outbreaks in the year in the next year. So it means that it's not necessary anymore to have new introduction. It doesn't mean that new introductions are not important, but it means that there is already overwintering of the lineages that can fuel new outbreaks in the next year by itself, not requiring new introductions from other states. And that's basically the same thing we detected with dengue 1. So for dengue 1, we detected genotype 5, which is the most prevalent all over Brazil. But in this particular state, we detect 4 clades with year-round transmission, sometimes for 2 years. The same clade was detected in 2 years in a row, right? So again, showing that new introductions may be important, but they are not required to fuel new outbreaks in the southernmost state of Brazil. So I also brought these papers here, this and the next one, to highlight to you the DENF3 virus introduction in Brazil after a 10-year gap, because as I mentioned to you in the beginning, understanding the expansion of those arbovirus in the southern part, and the subtropical part of the country, and also in South America, should be linked with the whole picture in order to provide a complete understanding of the distribution of those viruses. So dengue 3, the serotype 2 was not detected in Brazil since 10 years ago. And recently, we from the Fiocruz Genomic Network, led by Dr. Felipe Navega from Fiocruz Amazonas, we characterized the introduction of dengue 3 after this 10-year gap. So this is quite concerning, because now we have, again, serotype 3 circulating. And because of this 10-year gap, we are really, really concerned that the population immunity to block or to buffer the transmission of the F3 serotype is not there anymore because of this 10 years gap, right? So it's very likely that in the next month or next year, we're going to see a substantial spread and increase in dengue cases due to dengue-3 serotype 3, right? And another thing, another study that's help us to understand this of this larger picture of South America and Brazil comes from this recent study where we we sequenced 237 genomes of dengue-2, serotype 2, genotype cosmopolitan, and we detected multiple introduction and counter-wide spread of dengue-2 genotype 2. So with this study, we characterized at least 4 introduction into Brazil from Peru. It doesn't mean that directly Peru, because we have a lack of sampling in other countries. But what happens that in 2019 and 2020, there was a huge outbreak of dengue in Peru, but very few data from the specific serotype and genotype were available. But after this, we detected this dengue-2 genotype 2 cosmopolitan being introduced at least 4 times in Brazil and spreading countrywide. So we detected from north to south. So but why these examples are, yeah.
I think you can summarize your finding.
Okay.
Yeah.
Sorry.
Thank you.
Sometimes I speak too much. So I think I'm gonna go to the, yeah, just I'm almost in the last slides. Here I just would like to give you an overview about this huge need for global surveillance of arboviruses. You just saw that how important it is to have cross-sectoral and cross-country collaboration to understand the spread, because those viruses does not respect political borders. So we launched the APBioNETTalk inside of GISAID to bring the community together and to share the data, genomic data for arbovirus, to understand how they spread and what are the consequences, right? Here are some of the slides from GISAID. And I'd like to again highlight here that in the south of Brazil, we have these eroding climatic barriers. So we have larger transmission seasons, which are allowing these large outbreaks to take place in that part of the country where it did not happen in the past. So here are the take-home messages. Arbovirus burden in Brazil is increasing due to the circulation of new lineages. Recent increasing incidence is more pronounced in the subtropical region. There is a combination of factors that are leading to this epidemiological change, like arrival and communitary establishment of the virus, lack of immunological barrier of the population, and large population of the vector. As I showed, timely genomic surveillance of arbovirus can provide critical and detailed information to guide mitigation efforts, besides generation new insights, and robust and easily deployable bioinformatics tools are required to maximize the impact of genomic surveillance on public health. With this, I'd like to thank you very much for this opportunity. Sorry for, I speak too much today, but it's, as you can see and imagine, all this work is done by many hands. I have a large group from the Fiocruz Genomic Network, and also my inner cycle inside the Fiocruz Pernambuco and BNITM. And I also would like to thank you, the funders, for all the support so far. Thank you very much, and open to questions.
Yeah, thank you very much, Dr. Gabriel. It's a very interesting talk. So I'm also afraid to stop you because we would like to hear more. So anyone's, if you have any question, you can type, or you can just direct us, Dr. Gabriel's. And for the time being, I just wonder, okay, I saw your pipeline is very interesting, Dr. Gabriel's. So when I see in the pipeline, I mean the overview of the pipelines, the pipeline is actually use a reference genome assembly, right? So does it can apply to the unknown viral genome? I mean, to the unknown viral genome?
So yeah, it's not suitable for discovering new viruses. What the main use of viral flow is when we know something about a given virus. For example, we can sequence like metagenomic sequence some sample, a given sample, and then we detect a given virus using other methodologies to characterize highly divergent viruses. But as long as we have a reference genome, we can make this new reference genome, right? Annotate the genome of this highly divergent virus, and then we can use it inside of viral flow. So viral flow does require a reference genome where we know at least a little bit about this virus, at least the genome sequence and where the genes are.
Okay, thank you very much for the clarification. So we have a question from Manas Vini. Good evenings. My question is that what future developments or advancements in technology are crucial to further leverage these tools effectively?
So I'm not sure if I completely understood. What are the needs?
Future developments and advanced in the technology.
Okay.
Yeah.
Yeah, I think that's my take from my view from this question. I think the sequence technology is already there. We surpassed the bottleneck for sequencing. Of course, sequencing capacity is not equal all over the place. There is still room and big room to advance it in low research settings, how to implement those machines and those consumables to reach specific place of the world, which are the ones, many of those are highly impacted by arboviruses. But overall, I think the sequencing bottleneck was surpassed, because now we have miniaturized machines, we can take it to anywhere, and we can still generate a lot of data. From my view, I think the main bottleneck is the analysis bottleneck. And that's why we are trying to solve, or at least to bring some solutions with those pipelines to facilitate the analysis, because now we have plenty of data and we need to analyze this data in a timely way in order to generate results that will generate information, actionable information. So that's the main issue from my point of view. And you just mentioned your first question regarding divergent viruses. I think for genomic surveillance of non-viruses, we already have the tools. We already have the speed needed to report the results in a timely way. But for viral discovery of highly divergent viruses, we still are behind. We need to have better tools, tools that generate reliable results of highly divergent viruses, which is a really big challenge, because if we know very little of the virus sphere, it's really hard to fish those highly divergent genomes. But that's where I think we need to put our effort in order to make this information from metagenomic studies readily available for public health intervention, for example.
Okay, thank you very much. So we have another question from Amaresh Mishra. How does the use of bioinformatics contribute to the early detections and monitoring of the arbovirus outbreak? And what the challenge has been encountered in this process?
So yeah, thank you for the question. Regarding arboviruses, it's important to take into account the different biology and host and pathogen biology of these different viruses. One thing is implementing genomic surveillance for a respiratory virus, right? Another thing is implementing genomic surveillance for arthropod-borne viruses. Because for arthropod-borne viruses, we also have the vector. We can perform surveillance and also genomic surveillance focused on the vector, right? So I think the main challenge for arboviruses is first thing, how we can implement effectively genomic surveillances focusing on the vector. Why it's so important? Because sometimes we can detect the pathogen circulating in the vector before detecting huge outbreaks in the human population. So if we can detect the pathogen and characterize the lineage in detail before huge outbreaks, then we have an information that's critical for maybe mitigate or block the transmission of this virus in nature, in natural conditions. So I think that's one thing. Another thing is that for arboviruses, well, that's also common for respiratory viruses. We have a lot of asymptomatic patients. So we have a very narrow view or almost lack of information regarding which lineages are circulating in asymptomatic patients. If you find a way to perform surveillance on a broader scale, and also sample asymptomatic patients, maybe we will have more information that will be crucial for intervention. So I think, yeah, we need to think about specifically about each virus and how they are transmitted in order to evaluate which are the bottlenecks that we need to address for arbovirus, for instance.
Okay, so we have another question from Sharanya Monaharan. Great thought, Gabriel. Thank you so much. My question is, how is it important to address these minor variations in viral genomes, since it is very rapid to infect many hosts and undergo evaluations? The second question is, what do you think about how far this workflow has achieved? And the third question, what technique has been focused to address this issue?
Yeah, okay. Let me see if I can remember all of them. But the first one for minor variants.
Yep.
Okay.
Minor.
Regarding minor variants, that's intra-host evolution is something not only scientifically interesting, because we can see how the virus is changing inside of the host. In different tissues, for example, we can detect different minor variants. But coming from this huge amount of literature that was generated from COVID-19 pandemic, we can see that characterizing minor variants can lead us to detect specific lineages or mutations that will be fixed later on. So there are 2 or 3 papers showing that specific deletions that emerges in those VOC lineages, those variants of concern lineages, could be detected into the minor variant portion of some patients. So with this information of the minor variants, we could see how the virus is evolving and how and maybe detect which is the next step that could emerge as a new lineage and then replace the other lineage in nature. So I think the minor variant portion give us more detail about how the virus is evolving and how it may evolve in different circumstances. Another perspective from the minor variants that's interesting is that most of these minor variants variation in the same patient, for example, came from studies from patients that were infected for months or even years, right? Usually those patients are immunological compromised patients. So the virus can keep replicating for months on a row. And on those patients, we could, several authors characterize the minor variant portion, and they could see huge variation in terms of deletions, mutations, not only synonymous mutations, but also non-synonymous mutations that generate amino exchanges. And then some of these mutations, deletions and amino exchanges were detected as very important changes in some specific VOC lineages that dominated some period of the pandemic. So yeah, so summarizing this, I think minor variants are amazing to deep understand, because when we generate a consensus, a majority consensus, we are just simplifying the full range of information that's available from next generation sequencing. And we can have a much more detailed view if we look at the minor variants. And just the last point about minor variants, because that was kind of unexpected consequence from developing viral flow, is that when we reconstruct two versions of the viral genome, one of the major variants and one of them with the minor variants, eventually we started to detect co-infection events. So we saw that with this approach, we could easily spot some patients that were co-infected by two different SARS-CoV-2 lineages, but also from two lineages of chikungunya virus and also two lineages of dengue virus. We are detecting it systematically. So the minor variants portion, in this case, is not minor variants in the sense that it's generated de novo during the replication of the virus, but just because it looked like minor variants because we have two viruses co-infecting the same patient. And then we detect it in the minor portion because one virus will have lower abundance than the other virus, right? So that's another way we can look at the more, with a more realistic view about the virus evolution in natural conditions. So I think I only answered the minor variant thing. Which are the other questions?
Second question is, what do you think, how far this workflow has achieved? How far the minor variants analysis has achieved?
Yeah, that's very interesting. In fact, I showed to you one picture regarding the use of viral flow in Brazil, which for me was quite surprising and reassuring to see how it is being used all over the country. But I also have some data, not systematic collected, but data showing that people from the US, people from several other countries from South America are using viral flow, and also other countries across the globe. So it's important to say that other pipelines are as good as viral flow, probably, and some of them we tested and they are good. But it's really reassuring to see that viral flow, because we thought about developing viral flow in these containers, which make things easier for the end user, and also because viral flow was made to run very rapidly to generate these systematic and well-curated data for public health intervention. It has been used quite a lot. So I think, yeah, from my point of view, I think those are the main features that made viral flow widely used in the community.
Okay, so what technique has been focused to address this issue?
Sorry, can you repeat?
What technique has been focused to address this issue? What technique?
Yeah, but which issue? I'm not.
I think the issue
sure
is related to the minor variation.
Mm-hmm.
Yeah.
Okay,
so yeah.
Yeah, in order to analyze the minor variants, we implemented several thresholds, very restricted thresholds, because there are several papers in the literature showing that if you look at the minor variants and if you allow some not so strict thresholds, we start to catalog the error rate, the sequencing error rate. And that's not what we want to analyze, right? We do not want to analyze the sequencing error rate from the sequencing machine, but we want to see what is really happening with the virus evolution and co-evolution with the host. So we implemented several parameters which are well known in the literature to characterize it systematically, and everything that we did in terms of validation with other softwares, it was quite robust compared with what we implemented into viral flow. Yeah, so we are very, very confident that the minor variants that are reported by viral flow is also reported by other softwares. But that's important to say that minor variants we characterize per site across the genome. And one thing that we do not do inside of ViraFlow, because it's highly computational demanding, is how to phase them. How do we know that this minor variant was in the same genome of this minor variant from the 5-line portion of the genome, right? So because we have short read sequencing, we do not have the same nucleotide minor variant in the same read to be sure that they came from the same viral genome. So there are quite a few papers and software developed for phasing, trying to estimate which how many genomic variations and which minor variants came from the same genome that was sequenced, but we did not implement that inside of ViraFlow. And I think the main reason is that it will take much longer to analyze this data. And because ViraFlow was made with the main goal of public health intervention, this is not critical for public health intervention, although it's very interesting for understanding virus evolution. So that's one thing we want to test in the next phase, compare with other methods, mainly comparing with the phasing method in order to reconstruct these different genomic versions using the minor variants. But one thing that would be very, very interesting in order to understand the presence of the minor variants across the genome and if they came from the same genome is using long read sequencing. Because using Oxford Nanopore or PacBio, then we can sequence the full viral genome and detect, well, these and these and these variations that were characterized as minor variants came from the same viral genome. And that's, I think, it's the main, the best way to characterize minor variants at the genome level.
Okay, that's very long. I hope it's answer, Sharana. So we have another question. Shreya asking, good evening, sir. My question is, if we find the fundamental genome sequence, can we engineer the infection nature of the virus permanently right at the source? If we have fundamental genome sequence, can we engineer the infectious nature of the virus permanently at the source?
Well, I think that's a very interesting question, but it's well away from my background to answer that.
Okay.
You know, engineering viruses is something, yeah. Unfortunately, it's outside of my my field of.
Okay. Okay. So the next is from
Sorry.
Sridini. Good evening, sir. My questions, are there any unique characteristic or feature in southern Brazil that make susceptible to arbovirus outbreak? Does this workflow apply to all arbovirus?
Yeah, starting from the last one, we can apply ViraFlow 1.0, the one I showed to you today, for every arbovirus, because arboviruses, yeah, as long as you have a reference genome, like from Japanese encephalitis virus, from RV/FV virus, you can pass to ViraFlow and then it will run the core analysis. Regarding which are the features from the population or environmental features that are allowing these outbreaks to happen in the south of Brazil, I think it matches pretty well with one of my final highlights, where I pointed out that's a complex mixture of features, in fact. So the first one is that now we have the vector invaded Brazil, and now more recently he also invaded the south of Brazil. Because of this climate change, there is more suitable environment for development of the vector, and we have hard data showing that Egypt is distributed all over the place. The second one is that the population in that part of the country was not impacted by dengue as the remaining part, as the tropical part of the country was infected. So the population immunological barrier of the southern part of Brazil is very weak. It's almost nothing compared with the immunological barrier based on natural infections of dengue in the past. So basically, most of the population is naive, is susceptible for new infections, right? And the last one is something I already addressed. I think now we are experiencing more large transmission seasons where the temperature and moisture is ideal for vector development, and then there is quite enough time for transmission of those viruses in those communities. So I think these three main factors are interconnected and are responsible together for these large outbreaks that are happening in the region.
Okay. Okay, so that one, we have another questions from Ragil. So my question is, based on your experience, what factors significantly influence the genetic mutation of arbovirus? Is this phenomena observed in other viruses as well? Thank you.
Thanks. That's a very interesting question. In fact, arboviruses are particularly interesting to study because those viruses should be able to replicate in a vertebrate host and in an invertebrate host, two hosts that do have completely different biology and different molecules. So I think one thing that's mainly differentiate arboviruses from other viruses that are only transmitted through birds or between humans, for example, is that you do have this double host selection pressure buffering the evolution of the virus. So the virus, if you think about, well, this virus is well adapted to replicate in mosquito, then it's deleterious to this virus to be well adapted to one host because it also needs the other host to complete the cycle. And that's the opposite also true, right? So if you require a vector, you cannot be highly adapted to one host, then the infection will not move on because the virus lineage is not well adapted to replicate in the vector cell and the vector mosquito. So I think that's one thing that differentiates arbovirus from the other viruses, speaking more broadly.
Okay, so we have a last question here. Good evening, Sir Gabriel. You said that about NextFlow framework. Can you say more about it? Is there any framework compared to this? The
Yes.
NextFlow framework. Yeah.
Yeah, NextFlow is a domain-specific language. It was developed first for bioinformatics, dealing with a lot of sequencing data, right? And dealing with highly repetitive analysis, like we do for genomic surveillance. We need to run the very same analysis for hundreds, for thousands of samples. So this domain-specific language, although it was developed inside of the bioinformatics field, you can use it for every kind of task, right? And NextFlow is very, very interesting because although, of course, as any other language, you need to learn the syntax. The syntax is a bit different. You can integrate any kind of language inside of NextFlow. So if you do have an R script, and you do have a bash script, and you do have a Python script that, and you need to use these different scripts in different languages at different points of your pipeline, you just call it inside of NextFlow, and NextFlow will easily handle it. And I think another nice feature from NextFlow is that it has a very nice community. I think that's something so important in bioinformatics, right? Because we have a worldwide community and that help each other to solve issues and implement new pipelines. So our experience using NextFlow is really great because not only because of what I already mentioned, but for example, NextFlow, when you run the pipeline, you can see every step of the pipeline being run. And depending on many samples, there is a number. Well, there are 48 samples. It's run for 3 samples already, and it's being updated in your terminal version, right? So you can see the follow up every step of your pipeline, and that's all came as a default inside of Nextflow. You just implement your script, and then your script will show up as a name, and then you can follow this script running in real time. So those are features that are amazing for pipelines. You can see, well, there's an error here or there, or everything is going on well. And yes, there are other comparable technologies. The problem is that I'm not sure if I know exactly the acronym. I think it's WSL, but it's another. Oh, there is SnakeMake. SnakeMake is something comparable with Nextflow, but it's, of course, it's another syntax, but the idea is the same. And there is another one that works with the Amazon cloud, but now I'm not sure about the name. Sorry.
Okay, thank you very much for very long explanation. But Gabriel's sorry, we still have a lot of questions, but we're running out of time. So everyone, if you have any question, you can reach out Gabriel by emails to Gabriel's.
Yeah.
And yeah, thank you very much, Gabriel, for the very interesting sharings, and thank you very much everyone for participations. And don't forget to give your feedback, okay, through the link that we provide in the chat. Thank you very much. Thank you very much, Gabriel. Thank you very much, everyone.
Thank you. Thank you
Have
for
a
the
nice
invitation
day.
again.
Okay.
You too. Bye-bye.
Bye.
Bye-bye.